In our last video, we had an overview of the process of gene expression, learned a little bit about the history of how gene expression was first identified, and also talked about some fundamental concepts for the process of going from a DNA sequence to a protein. In this video, we're going to talk more extensively about the process of gene transcription and the process of modification. We're going to go through the molecular details in all their gory details. Okay, so we'll take a look at the molecular components, and then we'll look at stages of synthesis of a messenger RNA transcript. And there are three major stages to that process, and then those stages are followed by post-transcription modification. And so those three stages are the process of RNA polymerase binding and initiation, then elongation, and then termination. And I'll also note that there's another quick animation that illustrates this process with a nice British accent that's linked in the lecture slides. Okay, so here's our molecular components. So this process is orchestrated by a single enzyme that's called RNA polymerase. So it's got the name, or it has the suffix ase, which tells you that it's an enzyme. It also has the word polymer in it, which tells you that it produces a polymer. And then it has the word RNA in it, which tells you that it produces a polymer of RNA. So this workhorse enzyme can basically pull apart a strand of DNA at a small location, and then it will join RNA nucleotides that complement one of the two DNA strands. And in the previous video, I mentioned that the strand that is used is referred to as the template strand. So again, it's going to generate a complementary strand to the template strand. And just like with DNA polymerase, RNA polymerase works in a 5' to 3' direction. But because it's adding RNA, it doesn't need any kind of primer. But it does have to have a specific starting sequence. So it requires that starting sequence to bind and get chugging along the DNA. So that specific starting sequence, that's a specific collection of A, T, C, and Gs, is referred to as the promoter region of the DNA. So that's illustrated in this little cartoon to the right from your textbook, just using a different color for those DNA strands. So that promoter region is part of the DNA that's the starting point for this RNA polymerase. And then from this point, this polymerase is going to run along the lower strand, the template strand, and it's going to build a messenger RNA transcript. So it's not a copy, it's transcribing something that's complementary. So it's going to produce an RNA transcript that is complementary to this template strand of the DNA. There's a little bit more language to this also, which is we refer to DNA that's in the direction of transcription as the DNA that is downstream of this promoter region. Okay, so let's take a closer look now at the binding and initiation step for transcription, so part A. So it's simpler in prokaryotic organisms than it is in eukaryotic organisms. In prokaryotes, it works just as I described for you. So the RNA polymerase recognizes the promoter sequence, hops on, and it gets chugging along, transcribing DNA. In eukaryotes, the process is a little more involved, and we'll talk about this more when we talk about the regulation of gene expression. So for eukaryotic organisms, it doesn't just take RNA polymerase. Instead, there's a set of molecules, usually proteins, that are referred to as transcription factors that mediate the binding of RNA polymerase. So these transcription factors have to be present and have to bind in order for RNA polymerase to also bind. So they're kind of a prerequisite for RNA polymerase getting to work. And these transcription factors bind to a crucial sequence of DNA that's usually referred to as the TATA box because it has a general sequence of TATA, and then we have these asterisks. Those could be either an A or a T, and then there's another A and then another wildcard as well. So here's an example of how these things are organized for one gene. So here's the promoter sequence. Here is a TATA box. Here's something else that is upstream that we'll talk about in a moment. And then in this picture to the right, we've got the downstream portion, which is where the gene is that's going to get transcribed. Okay, so here is a more sophisticated diagram that shows more detail for how all of these transcription factors interact to facilitate the binding of RNA polymerase and the initiation of transcription for a eukaryote. So the main thing I want you to hone in on for the moment is this lower picture on this slide. So this shows some of those general transcription factors and then RNA polymerase on binding and starting to open up DNA and getting ready to begin transcribing the gene that's downstream. One other interesting part of this is that there can be other things that also bind and come together to form this transcription initiation complex in eukaryotic organisms. And this is a fairly complicated, so we're going to come back to this and we'll revisit it later on. Okay, so first step is just binding and getting started at that promoter sequence. So the next component of transcription is the elongation phase. And in some ways this is very simple. Basically what happens during elongation is the RNA polymerase just keeps adding RNA nucleotides onto the 3' end of this newly made piece of RNA. And it's pretty fast at doing this. So it can do this at a speed of 40 nucleotides per second. And as it does this, the DNA helix actually reforms right behind the RNA polymerase. And so the RNA will just peel away from that DNA template strand as it gets produced. And part of what that means is that this RNA polymerase doesn't have to finish transcription before another RNA polymerase could also bind at the promoter region and start to transcribe. And if another polymerase does that, starts to produce more RNA, then there are going to be more transcripts, and what that means is that there's going to be more production of proteins from that gene. Okay, so then the last step of transcription is termination. And so since we're talking about termination, we're going to see termination appear again. So I've got the terminator here just to remind you that he'll be back. And so for termination, in prokaryotes, so in the bacteria, basically what happens is there's a sequence of DNA, a specific sequence called the terminator sequence. And at that sequence, the RNA polymerase pops off and the RNA winds up being released. So all it takes is a terminator sequence and the process of transcription ends. And so again, in prokaryotic organisms, this whole process is taking place in the cytoplasm, right? Because there's no nucleus. So for those prokaryotes, the process of translation can start straight away. And so I've got a couple of pictures of this process for you. On the right-hand side is a scanning electron micrograph showing you what this looks like. And then on the left-hand side down here is an illustration of what you're seeing on the right-hand side. So on the left-hand side, in this case, the two strands of DNA have been separated from each other quite a bit. So we've got one strand that's inactive, and then the other strand is our template strand of DNA. And the process of transcription is happening from the right side to the left side. So you can see there's these little blobs that are right along this piece of DNA, and those little blobs are RNA polymerase proteins, so doing transcription. And then the other thing that this is showing is we've got a whole series of other blobs hanging off of this strand of DNA. And these other blobs are actually ribosomes that are translating the piece of RNA that has just been transcribed by these RNA polymerases. So these ribosomes are really not very patient about going almost straight from the process of transcription into translation. And so these are going to start translating down at the tip of this piece of RNA, and then they're going to move upwards to translate this message into a new protein. So in eukaryotic organisms, so prokaryotes, just to recap, termination is a result of reaching a terminator sequence. And in eukaryotes, the process of termination is pretty similar, but with a slight distinction. So in eukaryotes, the RNA polymerase reaches a series of nucleotides called a polyadenylation signal. So that's the series that is highlighted here, AAUAA, in that template strand. And when the RNA polymerase reaches that signal, it keeps going for a little bit longer, for somewhere between about 10 and 35 more nucleotides, and then it pops off and finishes the transcription process. So it's similar, but not quite as abrupt as in bacteria. Okay, so that actually takes us through the process of transcription. So we have binding and initiation with the promoter region, and in eukaryotes with the transcription factors. We have the process of elongation in a 5' to 3' direction along the template strand, and then termination, either reaching the termination sequence or the polyadenylation sequence, and then ending transcription. So now all that remains is to talk a bit about post-transcriptional modification. So this is something that happens in eukaryotes that doesn't happen in prokaryotes. And in this cartoon here, this process of post-transcriptional modification is also referred to as a process of RNA processing. So there are kind of two major types of modifications that are made to RNA after the messenger RNA has been transcribed. So first thing is that eukaryotic messenger RNA will contain extra regions that don't wind up getting translated. So that includes a cap, what's called the 5' cap, a tail, what's called the poly A tail, and then there are also regions that are referred to as the 5' untranslated region and the 3' untranslated regions. And then on top of that, there will also be introns that don't get translated. Okay, so the first modification that gets made to the mRNA is to add that 5' cap and to add the poly A tail. And that modification, those are markers that are put on the mRNA that signal that what's here is a new mRNA that's now ready to head to the cytoplasm, right? Since the RNA has to be moved, there needs to be some way to tell, is this new RNA or what's the story here? So then, and also that during the process of translation, that 5' cap is an important binding site for the ribosome. We'll come back to that. So that's the first major type of modification that's made to messenger RNA, is adding the cap and the tail. The second type of modification is referred to as splicing. So a eukaryotic gene is not a continuous sequence. It's actually split up into a bunch of shorter sections that are separated by regions that don't get translated. So the sections that do get translated are referred to as exons, and the sections that don't get translated are referred to as introns. So in the process of splicing, those introns get removed. So you go from a longer messenger RNA that's a mixture of exons and introns to a shorter final messenger RNA that only contains the exons that are going to get expressed. And this process of splicing is accomplished by a group of proteins and RNA molecules that are collectively called the spliceosome. So these are made up of these little units that are called small nuclear ribonuclear proteins, or SNRPs for short. And they're a hybrid molecule composed of a combination of RNA and protein. And these can identify sequences at the ends of introns and bind to those sequences. And they bring those sequences together and they form a little lasso shape. And then they join together the exons. So these SNRPs are also referred to as ribozymes because they're a type of self-splicing RNA. So this whole unit is what actually accomplishes the splicing process. And this actually brings us to something really cool, which is that it's possible for there to be alternative splicing arrangements for a given gene. So in this picture, up top I'm showing you a DNA sequence, and in that sequence we know what particular sequences code for those edges between exons and introns, so we know which regions are target regions for being spliced. And so in the gene that's pictured here, there are 5 pieces that are identified as potential exons. So when this gets transcribed into RNA, all 5 of those sequences will be present. But then through alternative splicing, it's possible to get different final RNA messages. So for example, if all of the exons are included in the final message, we'll have a longer RNA, and that will be translated into a bigger protein. And then in contrast, if only 4 out of the 5 exons are included, we'll get a different type of protein. And similar thing, it depends on which of those alternative exons wind up in the final processed RNA. So this concept of alternative splicing actually challenges that idea of the central dogma that you go from one gene to one protein, because with alternative splicing, you can have one gene and you can actually get to more than one type of protein. Okay, so that takes us through the processes of gene transcription and modification, looking at all of the molecular details with a little help from my cat. And so in the next video, we're going to now look at the process of gene translation.